Hello, my name is Mirza Ahmed, and the article I chose was on the effects of fasting, refeeding, and dietary fat restriction on plasma leptin levels. The research was done by David S. Weigel and his team, and it was published in the Journal of Clinical Endocrinology and Metabolism. Before we discuss the article and get into that, I would quickly like to go over the gut brain axis, also called the brain gut and microbiota gut brain axis, because we didn't really get the chance to talk about it in class. The axis between your brain and gut is composed mainly of the microbiota, or like the bacteria and the microorganisms living in your intestines, the neurons of your nervous system, hormones, and your immune cells. This system is used in the maintenance of your body's internal systems to, affect homeostasis and informing the brain on what the gut and more specifically the microbiota are doing. The axis itself is bidirectional, so your brain can affect your gut, but your gut can also affect your brain. Here I have an example of how your gut affects your brain. The microbes in your intestines consume food that you can't break down and converts them into byproducts that we can consume. Your gut interacts with the microbiota and produces hormones to communicate with the CNS and the brain about what the conditions are and if the microbiota are producing what they need to be. The brain processes these signals and decides the next action. Therefore, the gut, in this example, is using hormones to influence behavior. Now, onto the article. The article itself is about the influence that fasting and the amount of fat consumption, or I guess lack thereof, has on leptin production. Um, Leptin is a hormone that's produced by adipocytes. Uh, it's a hormone produced by fat cells. Its function as a hormone is to communicate with the hypothalamus about long term hunger levels and energy storage. It also seems to play a role in the regulation of energy expenditure. Previous research has found that leptin is a primary communication pathway between adipose tissue and the brain. David S. Weigel and his team, in this article, hypothesized that other factors influence leptin production instead of only total amount of adipose tissue.